MC1, the power of bacteria in the fight against hydrocarbon pollution. Oil is essential to our society, but the environmental cost is exceptionally high. During its production and use, residues contaminated by hydrocarbons accumulate, representing a significant environmental liability. Despite the existing physical-chemical methods, their inefficiency and high cost have driven the search for more sustainable alternatives. In this context, a new study conducted by researchers from LBMG-UFRN, published in the Journal of Hazardous Materials, highlights the advances in using a microbial consortium for bioremediation. Hydrocarbons are toxic and persistent compounds in the environment. However, biotechnology offers a sustainable solution, bioremediation through microorganisms to degrade these pollutants. But there is an obstacle: no single species of bacteria can degrade all the compounds in a complex mixture of hydrocarbons. This is where the MC1 bacterial consortium from Microbial Consortium 1, developed by our researchers, comes in. Comprised of 4 species of bacteria, MC1 works as a team, each member contributing specific enzymes to degrade toxic compounds. In tests, MC1 demonstrated superior growth and complete oil degradation capacity compared to individual bacteria. The results of the transcriptomic analysis indicated the consortium's rapid adaptation to toxic compounds, with early expression of stress response genes and degradation of xenobiotics. The study reveals that these bacteria have specialized genes for breaking down toxic compounds, such as benzene and naphthalene, which are generally resistant to traditional treatment methods. Surprisingly, Acinetobacter baumannii was the most active species, while Bacillus valescensis, although less active, played an important complementary role. Some genes are more active when oil is present, helping the bacteria to adapt quickly to the toxic environment. One gene that stands out is alk-B-alkane 1 monooxygenase, significantly increasing its activity during the final stages of oil degradation. This metabolic flexibility reinforces the potential of MC1 as an efficient agent in decontaminating hydrocarbon-contaminated waste. The study highlights the importance of synergy, showing how these bacteria collaborate and compete, leading to a more efficient degradation process than any single strain could. MC1 not only significantly reduces industrial waste's oil and grease content but also meets regulatory standards, proving its potential for large-scale use. MC1 represents a milestone in developing biotechnological approaches for the sustainable treatment of oily industrial waste, highlighting the role of microbial diversity as a strategic resource in mitigating environmental disasters. However, the research does not stop. Better understanding the roles of the hypothetical proteins identified and optimizing cultivation conditions could lead to even more efficient bioremediation in the future. MC1 is paving the way for a cleaner and greener future, where the smallest organisms significantly impact the treatment of oily waste. The planet thanks you. For more details, read our article Unlocking the Transcriptional Profiles of an Oily Waste Degrading Bacterial Consortium in the Journal of Hazardous Materials. Produced by the Laboratory of Molecular Biology and Genomics, UFRN. Financial support from CAPES and CNPq. If you found this work interesting, like and share it.